Okay, here we have 4 test tubes, A, B, C and D, a starch solution and some amylase. We'll put some starch into each test tube. So all 4 test tubes have starch. We'll remove some of the starch just to make them equal. Right, 2 of the test tubes, B and D, I will add amylase. So amylase has been added to test tube B and D. A and C still have starch. I will now leave the test tubes in some hot water, which is approximately at body temperature, 37 degrees. It's a little bit warmer there, but it will cool as the test tubes are put in. So they should end up, after about 10 minutes or so, the average temperature for all 4 test tubes will be close to body temperature. So the water bath will keep them at around the 30 to 40 degree mark. The temperature is around 37 degrees, so all 4 samples have been kept warm for 10 minutes, close to body temperature. So test tube A, test tube D, test tube C, and test tube B. Now, we will test A and B with a drop of iodine. Stir them slightly. Test tube A has gone blue-black in colour because there's starch present. Test tube B has not changed colour. Even with a few more drops of iodine in both, blue-black colour, reddish colour. Even though we put starch in all 4, there is now no starch in test tube B, which had starch and saliva. Test tube A, which is starch only, has gone black. Test tube C and D, we will add some Benedict solution. Remember, test tube C has only got starch. Test tube D has starch and amylase. We'll heat both of these over a Bunsen burner. So we let them warm up on the Bunsen until they come to the boil. Test tube D is now turning red in colour. Test tube C has no change. Test tube C had starch only. The Benedict solution shows there's no sugar present. Test tube D had starch and amylase. The Benedict solution is turning to a brick red, showing there is now a sugar present. If we compare our 4 test tubes now, test tube A had starch only, it was tested with iodine. It went blue-black. Test tube B had starch and amylase. It stays reddish colour of iodine. It was tested with iodine. There is no sign of any starch present now. Test tube C had starch. It was tested with Benedict solution. There is no colour change. The blue colour is the same as it was when we put in the Benedict solution. Test tube D has starch and amylase and was tested with Benedict solution. The Benedict solution has turned red, showing sugar is present. So in conclusion, when we added the amylase, the starch was removed and sugar was formed. So amylase turns starch into sugar. This shows the action of an enzyme.